Thank you very much for the introduction, and thank you also for giving me the opportunity to speak and to present in front of this audience about something that is not only my profession, but also something that is very close to my heart. Pharmacovigilance at its best, risk management throughout the life cycle of a medicinal product, of course beginning with the very first into-man studies and then carried on throughout the life cycle of a drug while it's on the market. Why do I talk about pharmacovigilance and not about drug safety? Well, really, I talk about both, but I think drug safety and drug efficacy are features of a drug. It's not something that you can do. So when I look at what I actually do for a living, then it's pharmacovigilance and not drug safety. You may well ask, risk? What risk? What is she talking about? We have this wonderful drug that we want to develop, and it's going to help hundreds, thousands of patients to decrease their suffering, or it will even help to save their lives. So where are the risks? Well, recent history has shown us that there are quite a lot of risks throughout the life cycle of a drug, beginning very, very early in the development of drugs. You may have heard of TeGenero and their compound TGN-1412. And this is what happens if you don't know enough about the risks of your drug. You may end up in the lay press on the cover page, but not in a good way. TGN-1412 was a monoclonal antibody, and it was designed and intended to help patients with leukemia or with rheumatoid arthritis. However, what happened with the very, very first into-man study was that 6 of the 8 subjects that were administered this drug had systemic organ failure, multi-organ failure, and almost died. Only the 2 that received placebo did not have any symptoms. And that was despite the fact that the administered dosage of TGN-1412 was supposed to be subclinical. And it could only happen because TeGenero at that point in time did not know enough about the risks of their drug. So you may well think, I don't see any fins above the surface of the water, so there aren't any sharks. My drug is a beautiful little goldfish. But also when your drug looks like this, is beautiful, but also very dangerous, you may not yet see the fin above the surface of the water. So you just don't know whether you have a goldfish or a shark. So what needs to be done, even in the very first into-man studies already, is risk management. How do you do it, and what do you do? There are 3 steps, starting with the characterization of the safety profile, that what you already know. What you can deduct, for example, when you have the, stick with the example of the first into-man study, what you can deduct from preclinical data or from the class of substances that your drug belongs to. You characterize the safety profile of the medicinal product. And then you plan for pharmacovigilance activities to learn more, to learn what other safety features your drug has, which risks, and how can you increase the knowledge about the general safety profile of your drug. When you've done that, the next step is to plan for risk minimization. How can you keep the risk that you already know of as low as possible and make sure that those measures that you're planning actually work? And then when you're done with step 3, it's back to number 1. So this is not something that you finish doing, but it's a cycle that continues while the drug exists. When you're considering the risks, of course, you always have to weigh them against the benefits. If you have, for example, a medication against headache, you wouldn't want, if possible, any risks attached to this medication. If you have a treatment for leukemia, of course you know that there will be risks attached to this product, and you're quite willing to take them because the benefits will outweigh the risks in most of the cases. So risk management is not only about the risks, it's also about the benefits, and it's always the scales of the two, the evaluation, the balance of the benefits and the risks. Now, what are safety concerns? In a regulatory view, when you look at the regulatory authorities, there are only 3 possibilities, 3 kinds of safety concerns. There are important potential risks, important identified risks, and there is missing information. These 3 would be safety concerns. For example, the term quite frequently used in clinical trial protocols, adverse events of special interest, that doesn't appear anywhere in the regulations, in the GVP regulations, GVP standing for Good Pharmacovigilance Processes. Now, what is an identified risk? An identified risk is an untoward occurrence, an adverse reaction, where there is adequate evidence of an association with a medicinal product. This could, for example, be something that you have seen in preclinical trials and that was confirmed by clinical data. It could also be an adverse reaction in a randomized clinical trial, an adverse reaction that you see with the medicinal product that you're developing, and that does not occur, or if so, in a much less frequency in the comparator arm. It could also be an adverse reaction that is suggested by a number of well-documented spontaneous reports. So when the drug is already on the market, when you have spontaneous reports with a sufficient data quality, you could also say, okay, this seems to be an adverse reaction, this seems to be a risk associated with my drug. What's a potential risk? With the identified risk, you're sure. With the potential risk, you have some suspicion, but you're not yet sure. So this would be an untoward occurrence for which there is some basis for suspicion of an association, but this association has not yet been confirmed. For example, it could be something you have seen in the preclinical data, but you have not confirmed it yet by clinical data. It could also be that in your randomized clinical trial, you see an adverse reaction in both groups. In the groups with your medicinal product that you want to develop, but at the same time in the comparator group. There is a difference, but it's not yet significant. It could be a signal arising from spontaneous reports, but either the data are poor quality or it's not yet that frequent that you would say, okay, this is definitely a risk attached to my product. And it could also be that you have a medicinal product in a class of substances, and for this class of substances, a certain adverse reaction is known, but you have not yet seen it with your product. So then it's a potential risk, but it's not yet an identified risk. And what makes a risk important? An important identified or important potential risk would be one that has an impact on the risk-benefit balance. So if you think of the scale, It tilts the scale. Or it could have implications for the public health. Then it's an important risk. Missing information. Why is missing information a risk? It would be information about the safety of a medicinal product that was not available at the time when you evaluated the risks of your medicinal product. So you just didn't know. You didn't see the fin above the water. And you don't know whether it's there or not. Examples would be missing information for populations that were not studied in the clinical trial. One of them is pregnant women. Usually you never conduct clinical trials in pregnant women. It could be children, it could be renal or hepatic impairment in patients. It could be the elderly. Populations you have not studied in your clinical trials will create missing information. Which means even if there is a medical reason why you exclude certain patient populations from clinical trials, you may have to rethink. Can you really be sure that once the product is on the market, those patients that were excluded in your clinical trials really will not receive your drug? If there is a chance that they do, then you should collect data in these populations. So you will need to look at the situation from different angles. It may well be medically valid that you have certain exclusion criteria, but if you think about the later, about the already authorized product on the market, and the patients that will be treated with that product, it may be necessary to collect data in those patients that you now want to exclude in your clinical trial. So what are the tasks of a marketing authorization holder for a company that has a product on the market? The tasks are to constantly monitor the risks, those that you know, and also watch out for those that you do not yet know about, but that you may detect during the life cycle of the product. The marketing authorization holder has to take appropriate actions to minimize the risks of the medicinal products and to maximize the benefits. This could, for example, be a selection of the appropriate patient population and the communication about this selection. Thinking of a practical example during clinical development, you have a clinical trial. You have a carefully designed study design. You have assessed the pros and cons of the various study centers that you're using. Your study program allows for the operative efficiency, and the scientific advice that you received was all in favor of your plans. To add local knowledge to the design of your study, you have chosen 3 CROs that are acting on your behalf. So, all seems to be well, right? Think about a couple of questions. If you're really using 3 CROs for your clinical trial, do you have access to all the data? Do you have access to all 3 databases? If you have access, how can you ensure that you do proper signal management, that you can actually evaluate the data during the clinical trial when they are spread out over 3 databases? Can you establish frequencies of adverse drug reactions? Do you know how they code their adverse drug reactions in those CROs? Do you have those coding aligned? What about cutoffs and follow-up periods? And can you collate all patient information as required? I'm not talking about the clinical study report that you're going to generate of all of the 3 data sets. You will combine the data sets in the end and write a study report. And then, of course, you will be able to answer all these questions. But what about during the clinical trial? Because in some situations, you will need to make a quick decision. But on what basis are you going to make that decision? You will need to have proof that the thing here in the water is a shark, not a dolphin. And how can you do that if you don't have all your data together? I have been talking about clinical trials all along, so it seems like once the clinical study program is finalized and you are applying for marketing authorization, you have all your data from the clinical trials in one place, in one document, then everything is well known and analyzed. Well, I'm afraid not really, as history has shown us, for example, with Vioxx. This is only one of the very prominent examples. There are a lot of others, but I chose this one because it's a prominent one. Vioxx was a non-steroidal anti-inflammatory drug marketed since 1999 and taken off the market in 2004 when a study revealed that the drug more than doubled the risk of heart attacks and death. And by that time, more than 38 deaths were attributed actually to Vioxx. That was one of the reasons why authorities and regulators decided that one of the headlines in pharmacovigilance would be put into practice. I'm talking about the headline, "It never happened unless you've got it in writing." So let's put in writing the risk management for a clinical trial. And the document where you do that is the risk management plan. The risk management plan must contain the following elements: the safety profile of the drug, that what you already know, step 1 of risk management. An indication of how to further characterize the safety profile of the medicinal products concerned, step 2 of risk management. The marketing authorization holder or the applicant who wants to put a product on the market has to document measures to prevent or minimize the risks associated with a medicinal product. So when you know that there are risks, you should think about measures how to minimize them. Could be that you select a certain patient population, that you select certain circumstances, how to apply your drug, but you minimize your risks. You can't completely get rid of them, but you can minimize them. And at the same time, you should think about how to measure the effectiveness of these risk minimization measures. This all has to be captured in the risk management plan. And then there are post-authorization obligations for marketing authorization holders once the product is on the market. There are rare adverse reactions that you will not be able to detect during your clinical trial program. It occurs, for example, once in 10,000 patients. To have a chance, even a chance, to detect it, you would have to treat 30,000 patients. And no clinical trial, and not even most of the clinical trial programs, will include so many patients. So there will always be risks that you cannot detect during clinical trial programs, but you will detect once the product is on the market. So with this requirement, there are also implicit requirements, which include a description of what is known and not known about the safety profile of the concerned medicinal product, and also a certain level of certainty that you are sure that your drug actually works. Because what you're doing in the risk management plan is weighing your benefits against the risks. So you don't know how much you can put onto the scale, onto this risk side, if you're not sure about how much you have on the benefit side. That's why the risk management plan is also about benefits. And then an effectiveness measurement of the risk minimization measures that you're going to take. This is the structure. First, there's a product overview. Then there is a description of what you know about the safety profile of your drug. That's the safety specification. Then there's a description of how you want to further assess the safety profile of your drug. That's the pharmacovigilance plan. There are plans for post-authorization efficacy studies, if relevant, because you may have to reassess the benefits of your drug. Then there are the risk minimization measures, including an evaluation of the effectiveness of these measures. And then there's part 6, a summary, and that's the public part of the risk management plan that has to be written in lay language and will be published on the EMA website or on the regulatory authorities website if it's a local product. And then there are some annexes. Don't need the details of those. The safety specification, again, consists of 8 parts. And I'm going to go into more details about the clinical trial exposure and the populations not studied in clinical trials, and then, of course, the identified and potential risks. So the clinical trial exposure will include pooled data from all clinical trials. They will be stratified by age, gender, indication, dosage, racial origin, and it will also give some hint of how long the patients were treated in the clinical trials with the product. And then there are the populations not studied in clinical trials. They have to be described in the risk management plan, and it will give you the contraindications really for your product. Because if there are certain patient populations that were not studied in clinical trials, then there is really no basis to say this drug is safe and effective in these patients. So, this is the consequence of your exclusion criteria. What does it look like in the risk management plan? You list the exclusion criterion, and you say whether it has implications for the target population, and if so, which implications. For example, if you have an Alzheimer's study and you have not conducted any studies in children, there will be no implications for your target population because children don't get Alzheimer's. But if you have excluded patients with a chronic heart disease, then of course there will be implications for your target population because the elderly who get Alzheimer's also can have a chronic heart disease. And then there's a table looking something like that. You're listing the exclusion criterion, the reason why it was an exclusion criterion in your clinical trial, and if you think still this should not be a contraindication in patients treated with this drug, then there needs to be a justification for this. Then there is a module with the identified and potential risks, listing the newly identified safety concerns, any study reports with implications for safety concerns, details, and identified and potential interactions with drugs and with food, and pharmacological class effects. In the pharmacovigilance plan, when you have already a safety profile, you still need to identify any new safety concerns. How will you characterize them? How will you investigate a potential new safety concern, and what are you going to do about this missing information? Then the risk minimization measures, what are you going to do and how are you going to measure whether what you're doing is effective? And then the public summary of the risk management plan itself, publicly available in the internet, and listing not only the data but also the concerns about the study and further plans of what should be done about those safety concerns. When do risk management plans have to be written? They have to be submitted with any new drug application in the EU. They have to be submitted when there are considerable changes to a marketing authorization. For example, a new indication or a new route of administration, at the request of the agency being the European Medicines Agency, not FBI or NSA, and at the time of the renewal of the marketing authorization if the product has an existing risk management plan. So, at some point in time, all the drugs will have a risk management plan. All the drugs in the EU will have a risk management plan. So you can feel safe, right? You never know what's underneath the surface. So watch your shark. And I'm taking any questions. Thank you.